I recently read Richard Le Gallienne's Young Lives, clued to me by JC, which book described his younger life, loves, and his written words of poetry and prose, en route to his job as a columnist, novelist, and poet. His young romances began around the age of 20, these endeavors of love having been necessarily earlier restricted during the prudent Victorian era. What a difference 150 years makes. No more chaperones. Go back thousands of years, though, when the lifespan was only about 40, if that, and we'll find marriage at 13. So it is that nature still gets our bodies ready for procreation even by 12 now, and I heard that it may have even moved up a year. Gallienne became relatively famous. I'm not, but JC had pointed out some similarities, such as lineage, history, general outlook, metaphysics, romanticism, elfin lore, writing a rubaiyat, sailing ancestors, and poetry prose, not to mention the love of the female form. My reading of Gallienne's exploits spurred me to write my own autobiography, realizing that there were enough twists and turns of fate in my life to carry it forth, and from even younger times, plus there were ever the universal themes that a general audience could appreciate. Plus, I had a reservoir of untapped material. I'm 75 at the time of this writing, and I've perhaps exhausted the other genres, having done near one of each. Who really cares, though, about my life in particular? Probably not many, but I still had one of the easiest writing jobs, the retelling of actual happenings, and so a lot of words flowed out. Rather effortlessly, they practically writing themselves. I had to take advantage of myself while I was still in the mood. There are also the heights of emotion that sweep one over and through while writing. Our rememberings try to describe reality as it really was experienced, but that sheer essence may elude, although some general outline remains. Then too we add to it, subtract from it, and reconnect by association to the new. Lo, the subjective meets out our reality, while the objective lies furthest removed. How do some crumbs here and of the past waft back as vapors unto our present? Do the senses of smell and taste, yet more fragile and more insubstantial, bear a unique burden of memory, as more enduring and faithful, rising up past the ruins of the rest? Just noting the butterscotch back then, without its tasting, would not have made the mark. Time mutates some ancient pastimes, and so they are not wholly recaptured and sometimes rather fallible, even altered more by the call to mind, yet they are there. A memory begins as a changing connection between two neurons. The strength of the synapse changes so that the neurons can communicate. Thus, the taste of memory also activates the neurons downstream to do with one's childhood days. The neurons have been inextricably entwined, yet too reconsolidate upon recall. Jonah Lehrer has the full answer, summarized. The memory-making process needs proteins for the cellular construction of remembrance, yet the life of a protein is but 14 days. And some hippocampal neurons die, and some are born anew, yet some memory seems immutable. Does the mind constantly reincarnate? Aye, our memories must be made of a material stronger than cells, and must be quite specific as well. While each neuron has but a single nucleus, it has a teeming mass of dendritic branches, connecting to other neurons at dendritic synapses, such as the branches of two trees touching in a forest. So it is at these tiny crossings that memories are made, not in the trunk of the neuronal tree, but in its sprawling canopy. What marks a specific branch as a memory? What molecule awaits the taste of butterscotch? It has to turn on mRNA to help make the proteins. Its name is cytoplasmic polyadenylation element binding protein. A tough assignment of a name for even my memory to recall. So, how about CPEB for short? Since it was in the brain's memory center, scientists looked for it in sea slugs, amazingly finding it in the slug's neurons. Upon removing it, the sea slugs could not remember a darn thing. But how does it work, existing outside of time? Well, it has a series of repetitions in its amino acid repetitions, QQLQ, Q, BQL, QQQ, where Q is glutamine. Looking for similar odd repetitions, what looked like a prion was found. Prions are pathogens of Earth's nastiest diseases. They are everywhere and have two distinct states, as no other proteins do, one active and one inactive. Without guidance from above, they can switch states and alter proteomic structure without changing DNA, and then transmit their new infectious structure to neighboring cells with no transfer of genetic material. Biology's sacred rules are violated. In the brain, Krebs proteins are sturdy enough to resist time, they being virtually indestructible. Yet they have plasticity, being free of the genetic substrate, to change their shapes, creating or erasing a memory. When we think, the neurotransmitters serotonin and dopamine are released by neurons, which switch the CPEB protein into its active state by changing the very structure. The activated CPEB marks a specific dendritic branch as a memory, recruiting the requisite mRNA needed to maintain long-term remembrance. And yet, prions have an element of randomness built into their structure due to the inscrutable laws of protein folding and stoichiometry, even becoming active for no reason. Such contingency is just like Proust predicted. The remembrance of things past may not be the remembrance of things as they were. Due to unpredictable and unstable prions, we may have some essential randomness, for memory obeys nothing outside of itself.